Today I will be covering the cell cycle in eukaryotes. First, I will cover the importance of the cell cycle, and then the eukaryotic cell cycle overview. Next, I will talk about each of the phases of interphase, including the G1 phase, G0 phase, S phase, and G2 phase. Thirdly, I will cover the processes of mitosis, including prophase, metaphase, anaphase, telophase, and cytokinesis. I will cover the cell cycle regulators that control the transitions of the cell cycle phases, including cyclin-dependent kinases, also called CDKs, cyclins, CDK activating enzymes, and CDK inhibitors. Finally, I will review the important cell cycle checkpoints that regulate the cell cycle efficiency. Why is the cell cycle important? Cell division is vitally important for the survival of all organisms, and therefore life itself. This multi-step and heavily regulated process allows organisms to grow. Furthermore, cell division is important for repairing injuries by replacing cells that may be dead, damaged, or simply too old to function properly. In this manner, cell division provides a regeneration of new cells over and over to keep the body functioning properly. While this cell division rate is highly dependent on the cell-specific type, the human body has trillions of cells dividing each day. For example, brain cells are believed to not divide much, if at all, in a healthy human lifetime, while skin cells must divide quickly enough to replace over 30,000 lost every minute. The careful regulation of cell division is carried out by many cell cycle regulators. The cell cycle encompasses each state that a cell may be in any moment of its life. Mitosis is a phase of this cell cycle that involves the division of cells in eukaryotic organisms like humans. This process ultimately divides one mother cell, pictured here on the far left, into two identical daughter cells, pictured on the far right. The first process, labeled DNA replication, occurs during the S phase of cell division within the interphase process, which we will discuss in more detail in the interphase portion of the lecture. Next, the process marked cell division shows the simplified process of mitosis, occurring to divide the duplicated chromosomes between two daughter cells. As I mentioned, while this is an oversimplified illustration, it accurately depicts the duplication and movement of chromosomes during the cell cycle division of mitosis. The remaining portion of the cell cycle occurs between completion of the daughter cell formation and the beginning of DNA replication, and between DNA replication and mitosis. These two cell cycle portions are termed gap phases that will also be discussed in interphase. Together, each of these phases operates in a very specific manner to carry out the division of cells. In fact, this process occurs in more of a circle rather than a line that is pictured here. In this illustration, the orange ring represents the process of interphase. While the main concern of study is typically focused on the M phase of mitosis, represented by the yellow and purple M's, the cell resides in the interphase portion much longer than the actual division of the cell in mitosis. This extended interphase interval is necessary for cell division preparation and is much longer to ensure all the components needed for cell division are present, available, and without error. In the G1 phase, represented by the light blue, the cell is preparing itself for DNA replication that will occur later in S phase. This preparation is marked with organelle replication, growth of the cell, as well as RNA and protein synthesis. Furthermore, during the G1 phase, cells may enter the G0 phase, represented by the black circle leaving and entering the G1 blue phase. In this phase, known as cell cycle arrest, non-dividing and non-growing cells reside until they are signaled to return to G1 and resume the cell cycle. This is the location of most nerve cells, as discussed previously. On the other hand, some cells may rarely or never reside in the G0 phase, like skin cells, because their replication is so necessarily frequent that they do not arrest their growth or division. For a cell to move from G1 phase to the next process in interphase, known as the S phase, they must pass checkpoints set forth by the cell cycle regulators. For this particular transition, the checkpoints include cell size, checking for damage to DNA, nutrients, and growth factors. This checkpoint is important because all these components are necessary for accurate and efficient DNA replication. After the checkpoints of G1 phase are achieved, the cell enters S phase, represented by the red bar. During this phase, DNA synthesis results in duplication of DNA. RNA and protein synthesis also continues. Next, after the DNA replication is complete, the cell enters G2 phase. In this phase, the cell continues to grow, typically twice in size, and replication of organelles occurs in preparation for mitosis. Much like the checkpoint in between G1 and S phases, the G2 phase also has a checkpoint before the cell reaches the M phase of mitosis. This checkpoint evaluates cell size and any DNA replication errors before allowing the cell to pass through to begin mitosis. After the G2 checkpoints are reached, the cell can enter mitosis. The first phase of mitosis is called prophase. In this portion of mitosis, chromosomes condense, spindles appear from the centrosomes that move to opposite sides of the cell, and the nuclear envelope begins to degrade. This is depicted in this illustration with the spindle fibers appearing in the second and third cell as the black lines, and the centrosomes represented as the small blue lines in circles at the edge of the cell. The now visible and condensed chromosomes are represented by the red and green X's. The spindle formation is imperative for later chromosome attachment, and the centrosomes serve as the pulling forces behind them. After prophase, the cell enters metaphase. In this phase, the chromosomes, represented in red, align at the center of the cell, and the spindles extending from the centrosomes on either side attach to the sister chromatids. The spindles are represented in blue and the centrosomes in orange. The spindles attach to the sister chromatids via the centromere in the middle. Next, the third step in mitosis is anaphase. During this phase, the sister chromatids become individual chromosomes by splitting and are pulled toward the ends via the spindles and the pulling of the centrosomes. Finally, during telophase, the chromosomes decondense on each side of the cell separately. New nuclear envelopes form around each separate nucleus on either end of the cell, while the fibers break and continue to push the ends apart. The final step of mitosis involves the breaking of the two new cells into their own individual cells. This is called cytokinesis. After this step, two new identical daughter cells individually enter the G1 phase of the interphase cell cycle. This starts the entire cell cycle process over again. This illustration brings each step together with their key purpose. Interphase is responsible for DNA duplication. As we discussed, the most important aspect of this replication is accuracy. Next, Prophase is important for nuclear envelope destruction and condensing chromosomes. Metaphase is key for spindle formation. Anaphase separates and pulls the chromatids to opposite sides of the cell. Telophase forms two nuclear envelopes for each nucleus. Finally, cytokinesis separates the two identical cells. Cell cycle regulators are the driving forces behind cell cycle completion. These regulators are responsible for moving cells through the cell cycle stages. In fact, cells can only complete the cycle and undergo division if they undergo cell cycle regulator influence and pass checkpoints that ensure DNA damage has not occurred. Cyclin-dependent kinases are one of these regulators. Also known as CDKs, cyclin-dependent kinase levels remain consistent during the entire cell process. In contrast, the other cell cycle regulators, called cyclins, have varying levels throughout the cell process. This is because the cyclins are proteins that activate the CDKs. Therefore, their levels cycle up and down throughout the cell cycle to activate the CDKs at the appropriate time. Starting in the yellow G1 phase, cyclin D is necessary for cell entry into the first gap phase. Cyclin D binds to cyclin-dependent kinases 4 and 6 to activate them. As with all the cyclin CDK complexes, this activation of CDK results in protein phosphorylation. This phosphorylation cascade, started by cyclin D, CDK4, and CDK6, allows the cell to enter the G1 phase. Cyclin D is produced only when stimulation of growth factor occurs. The next regulator is in the orange region between the yellow G1 phase and the red S phase. When cyclin E associates with CDK2, the cell progresses to the S phase. At this point in the cell cycle, cyclin A binds CDK2, which is necessary throughout the S phase, as well as the transition to the G2 phase. At this point, cyclin A forms a complex with CDK1 to ensure mitosis or M phase entry by the cell. Finally, cyclin B complexes with CDK1 for mitosis regulation. Each of these complexes is important for progression of the cell cycle. CDK activity is dependent on dephosphorylation by CDK activating enzymes. These enzymes are in the CDC25 family. For the G1 to S phase transition, CDC25A is the important enzyme. CDC25B is an S phase activation enzyme, and CD25C aids in the CDK1 cyclin B complex activation during mitosis or the M phase entry. These enzymes are represented in this illustration as the activating phosphate present on the red CDK in the active state, shown in the pink. On the other hand, the Wee1 kinase, represented by the inhibitory phosphate on the red CDK in the inactive state, represented in gray, can inhibit the CDK cyclin complex action. This is yet another method that can prevent inappropriate movement through the cell cycle, like premature mitosis. Finally, CDK inhibitors can also inhibit the CDK cyclin complex. This suppression of growth is achieved through PRB activation, which results in the inactive complex represented in the illustration at the bottom with the dark green C shape over the CDK cyclin complex. Growth suppression is an attractive mechanism of CKIs, which could be useful for the suppression of tumor growth. As we have discussed, checkpoints exist throughout the cell cycle to ensure proper and precise duplication of cells. First, the checkpoint between G1 and S phases is illustrated by the purple bar in the bottom right of the cell cycle circle. As mentioned, cell size, nutrients, and the signals from CDK cyclin complexes are necessary for progression. However, the most important halt at this step is making sure DNA is undamaged. Damaged DNA is dangerous for any cell, and the body has these checkpoints to prevent the replication of damaged DNA. The checkpoint between G2 and M phases also has this requirement, represented by the purple bar at the top left of the cell cycle circle. In both cases, the cell can be blocked by a protein called P53 when damaged DNA is detected. This gives the cell an opportunity to repair the DNA before continuing the path to division. In fact, damaged cells have increased levels of P53 to make this failsafe easier. Ultimately, if the damage is too severe to repair, P53 can cause apoptosis or cell death to prevent future cells from possessing the damaged and dangerous DNA. This extremely detailed image shows the complicated cascade of activators, DNA damage detectors, and their relation to the cell cycle. Starting on the top right, NF-kappa B is required for cyclin D expression. As discussed previously, the cyclin D CDK4 complex is important in the G1 phase. Specifically, this complex causes the hypophosphorylation of the RB protein pictured in green. Then, the RB protein interacts with cyclin E CDK2 complex pictured in brown for hyperphosphorylation to result in the PRB, also pictured in green. This interaction with cyclin E CDK2 elicits the expression of cyclin A, necessary in the next step of the cell cycle, known as the S phase. Returning to the top of this illustration, all of these steps may be inhibited in the presence of I-kappa B, which can block the action of NF-kappa B through proteasomes. Also, important in the G1 phase is P53 activity. First, this activity can be inhibited when necessary by MDM2, represented in the little gray circle. However, when DNA damage is detected, P53 is a vital cell cycle tool. First, when a break in DNA occurs, it is detected by kinase sensors. These sensors are labeled ATM and ATR, represented by the purple circles on the right-hand side of the illustration. Specifically, ATM detects breaks in double-stranded DNA, and ATR detects breaks in single-stranded DNA. These signals set off the signaling cascade through checkpoint kinases. These kinases are denoted CHK1 and CHK2, also represented in purple. At this point, CHK2 activates P53, which in turn activates P21. This activity inhibits CDC25A, which is the necessary phosphorylating enzyme for the cyclin A, CDK4, and CDK6 complex, as well as the cyclin E and CDK2 complex. Furthermore, CHK1 directly inhibits the CDC25A enzyme. The WEE1 enzyme inhibitor is also depicted at this step, which can exclusively block cyclin E CDK2 complex. This multi-step and complicated process serves to halt the G1 phase to give the cell the opportunity to repair DNA damage. As discussed before, P53 can aid in apoptosis or cell death if repair is not possible. This DNA damage checkpoint is located between the G1 and S phase and represented by the red circle at the bottom of the right of of this cell cycle image. In a similar fashion, in G2, ATR activates CHK1 to directly inhibit CDC25C, while ATM activates CHK2 and continues the cascade to activate P53 and then P21. This in turn inhibits the cyclin B CDK1 complex that CDC25C typically phosphorylates for activation. This is another DNA damage checkpoint represented by the red circle on the left side of the cell cycle in the G2 phase. Furthermore, another checkpoint is present within mitosis. This checkpoint is represented by the orange circle within the purple M phase of mitosis. Known as the spindle assembly checkpoint, or SAC, this point in the cycle halts the activity of mitosis if the spindles are not aligned properly in metaphase. Specifically, this checkpoint involves the delaying of anaphase so chromatids are not split incorrectly or incompletely, resulting in daughter cells with excessive or lacking chromosomes. Overall, while complex, this illustration gives a detailed and full picture of the complexity that is cell cycle regulation. In conclusion, the heavily regulated and unique specificity of the cell cycle is responsible for the survival of every organism in the world. Without cell cycle replication, not only would cells not be able to repair, replace, or grow, but human existence as we know it would be obsolete. After all, each of us started out as one simple cell that had to divide on a massive scale to produce the functioning organism that is a trillion-cell human being. Furthermore, the careful regulation of this cell cycle by regulators and checkpoints attempts to combat the ever-pending danger of disease and cancer that can result when damaged DNA is permitted to survive and replicate. So the cell cycle is not just an important concept for survival, it is the only way for survival.